Good morning to everyone. I hope I am audible to all of you. So thank you all for joining us today. My name is Himashri and I am a senior executive at the for Science Communications at the DBT Wellcome Trust India Alliance, and I will be your host for today's lecture. So today we bring to you the third lecture of the continuing biotechnology education lecture series, and this lecture series have been organized by India Alliance in collaboration with Guru Angad Dev Veterinary and Animal Sciences University and IIT Roorkee. Our aim is to bring to you a diverse range of topics in biotechnology. The first two lectures of this series focused on genetically engineered vaccines and the next generation sequencing. And today our topic is metagenomics. So we welcome all of the participants who have joined us from GADVASU, IIT Roorkee and lots of other institutions around the country. So let me start with a quick introduction to the organization I represent, the DBT Wellcome Trust India Alliance. India Alliance is a charitable trust registered in India that funds research in health and biomedical sciences. So yes, just like the DST, the DBT, the CSIR that we all know, we also do fund research in India. But apart from investing in these transformative ideas, we also support research ecosystems to advance discovery and innovation to improve health and well-being. We are being funded by the Department of Biotechnology, Government of India and the Wellcome Trust, UK. Apart from this, we also fund various programs that promote various national and international collaborations, professional skill building workshops, and also programs on strengthening research ecosystem, and we promote science education, awareness and public engagement with science in India. So India Alliance conducts such workshops and lecture series from time to time, and we encourage you to keep engaged with updated on our fellowships, research grants and other events by following us on social media. We'll show you the handles on the screen. Next slide, please. Okay, so you can see some of the handles are for our Facebook, Twitter, YouTube channels are there, and we regularly post updates on there too. So please keep us followed. So before we dive into the lecture today, a few housekeeping matters. Please note that this webinar is being recorded and we will be sharing snippets of it on the social media handles. Your microphone and videos will remain switched off during the event to avoid any distractions. You can leave any questions you have for the speakers anytime during the session in the question box. In between, the speakers may ask certain questions, at which times we will unmute you for the short duration. You also please make sure that the questions only relate to the topics being covered in the webinar. If you have any other queries, we'll share email IDs with you that you can write to us at. In case of any webinar, in case of any technical glitch, and if you find that the webinar ends abruptly, please join back using the same registration link. Please also note that we'll be providing e-certificates to all of you who attend the entire session and share a short post-workshop evaluation form with us. We'll mail it to your registered IDs within 6 to 7 working days. And if you plan on sharing highlights from the workshop through your social media handles, you are welcome to do so. Please feel free to tag us. So next slide, please. Yeah, so now we come to the agenda for today's workshop. You can see that we are starting with the introduction, and now we will have a welcome address and an inaugural address, and then we'll start the first session on metagenomics, and this will be followed by a session from India Alliance on funding opportunities available at India Alliance and a question and answer session. So let us come to Dr. Jyotsna Dhawan. She will be introducing India Alliance to you. She is the CEO of DBT Wellcome Trust India Alliance, And to briefly introduce her, she is a cell and developmental biologist who has worked on stem cells and tissue repair. She has led a group at the Center for Cellular and Molecular Biology, (CCMB), and was involved in the establishment of DBT's Institute for Stem Cell Science and Regenerative Medicine in Bangalore. She has been actively involved in mentoring young biologists. So with this, I hand over the screen to Jyotsna for her speech. Thank you. I think Dr. Jyotsna has um, internet issues. So can we just go ahead with the introduction first for GADVASU, and then by the time we'll be back with Dr. Jyotsna. So, we are supposed to have Dr. Yashpal Malik from GADVASU for the talk, but I have just received the message that he is not available due to some reason, and I'll now request Dr. Ratan Chaudhary to give a brief introduction.
Okay. Good morning to all. So this is, as we all know, biotechnology is an exciting and ever-expanding field of science that harnesses cellular and biomolecular process of living beings, microbes, animals, and plants, to develop a usable product of for mankind and society. The big aim of DBT, Government of India, is to make a country 100 billion bioeconomy by 2025. Since India has more than 200 biotechnology industry and Punjab is ever growing, the biotechnology and pharmaceutical industry by establishing agri-food biotech clusters. For example, in Mohali, we have Mohali Biotech Park and many other biotechnological incubators. In addition to that, Punjab has 11 medical institutions that supports life science program. So the objective of this training program, Continuing Biotechnology Education, CPE, is to strengthen the biotechnological skills and provide the updated information of biotechnology to the students, staff, researchers, and professionals, and to build a scientific network of students and professionals for their mutual benefits. The aim of CBE is to provide a learning skill-based techniques, provide familiarity with cutting-edge technology, equipments, and updated research-based knowledge of biotechnology. So this is the third lecture series, and today we'll be hearing Dr. Mukesh Gupta. So with that, thank you everyone, and I would like to thank DBT, my Vice Chancellor, Dr. Inderjeet Singh, and a whole team of DBT India Alliance. So thank you very much, and we are looking forward to hear today's speaker, Dr. Mukesh Gupta. Thank you. Over to DBT.
Thank you so much. I'll just have a quick check if Dr. Jyotsna has been able to join.
Thank you for your patience, everyone. I just got word from Dr. Jyotsna that there's a problem with her system network. So she's recommended that she will give her introduction before Madhavan's session on funding opportunities. So we can have Dr. Mukesh Gupta have his session right now. Yeah. Thank you, everyone, for your patience. Bye-bye.
Thank you so much, Nicolette. Uh, so now we move on to the next and the main session for today. So first of all, I'll introduce Dr. Mukesh Gupta to you all for the moment. So Dr. Mukesh Gupta is a professor of biotechnology at NIT Rourkela. I'm sorry for the mistake on the slide. He's a professor of biotechnology and mechanical engineering at NIT Rourkela. He has done his bachelor's in veterinary and animal health and a master's in veterinary science, and with gold medal and top of his class in both of them. He has completed his PhD from the Konkuk University in South Korea, and he has a long experience of more than 18 years in academic teaching in various institutes of the country. He has also taught in Konkuk University, South Korea. So now, and also he has been the recipient of numerous awards, recent, and one of which is the recent fellowship by the National Academy of Agricultural Sciences. So we congratulate him on that. And he has also 14 patents to his name already, and many patents are waiting, and so there will be more to his name now. And he has over 200 publications in reputed journals, in international and national reputed journals all over. So now with this brief introduction, I will hand over the screen to Dr. Gupta for his lecture today.
Thank you. Thank you to the India Alliance and GADVASU and IIT Roorkee for having this opportunity to share my thoughts and views on today's topic, which is on metagenomics. Can you please just make me the presenter?
Dr. Mukesh, you have been invited.
Okay, so let me share my screen with you. I hope my screen is visible to all.
Yes, Doctor.
Okay. So I'm going to talk about the metagenomics. Some of you may be familiar, some of you may not be familiar with this topic. So I'll start all about from the basics of the microbiome to measuring and interpreting the microbiome. That's all this topic is all about. So whenever we talk about microbiome or microorganism, I think the first thing that comes into the mind of the people is bacteria, the diseases, the difficulties in treating the diseases. And I also recall back on my days when we used to have several cases of complex diseases or wounds which were very, very difficult to treat, and we had excellent facilities to diagnose them. We have excellent facility to even find out what antibiotics they will respond to, and we used to treat them. But we have horrendous tasks of treating them, and well known to all the veterinarians, well known to all the medical professionals, that how difficult can be treating certain wounds or certain diseases such as mastitis, which is it's a national problem, treatment of mastitis, where the animals or the humans doesn't respond to the antibiotic. Even if we select the antibiotics using antibiogram, we fail to treat the disease with antibiotics, and it remains a challenging task to the veterinarians worldwide. It remains a challenging task to medical professional to treat the bacterial diseases with the available antibiotics. Now, thanks to metagenomics, today we are in the era of science where it is possible to profile all the microorganisms that are present in a particular tissue sample, may it be wound, may it be blood, may it be milk, or may it be any other biological fluid, and devise our proper therapeutic strategy for treatment of that particular disease, or to find out novel therapies that can be used for it. So both start from diagnosis of diseases, prevention of diseases, to treatment of diseases. And that's what we are going to talk today, is metagenomics. It has made something possible which was not possible earlier with the traditional methods of diagnosing or traditional methods of microbial culture or microbial profiling. So today we are in that era of metagenomics, and metagenomics is not just about profiling the microorganisms for diagnosis or for treatment. It goes beyond that. It goes beyond to even diagnose, if knowing about the metabolic syndromes, obesities, and other physiological and pathological phenomena. Say, for example, you may be surprised to know that you may have seen people around you who eats less but tend to gain weight a lot and become obese. People like me, I eat less but become obese. On the contrary, you may have also seen many of your friends who ate very, a good amount of assumption food, but they still remain very thin, and we feel very jealous about them. And we blame it to our genetic makeup, we blame it to our lifestyle and so on. You'll be surprised to know the way back in 2005, 2007, people found that this whole obesity and metabolic syndrome could be linked to your microbiota, microorganisms, bacteria that are present in your gut. It's very interesting to note that, when they did the profiling, the Eckerberg's group, the Elizabeth Biggs group, they profiled the microorganism of the obese people, the gut of the obese people, and the gut of the lean people, and they found that the metagenomic analysis of this organism, that the profiling or the type of microorganisms that are present in both individuals are quite different. So the obese people had a lower diversity of gut microflora than the lean people. Obese people have reduced diversity, and particularly the vermiculates group of bacteria were predominant. Gram-negative bacteria were predominant. These microorganisms can damage the gut epithelium, can produce lipopolysaccharides, which can pass through the gut epithelium, can go into the blood, can activate inflammation, local inflammation, as well as can, beyond causing the inflammation, they can also include insulin resistance. They can go to the blood, stimulate the TLR4 activity to induce the transcription of cytokines, which may affect the MAP kinase pathway, JNK pathways, and so on, to activate the NF-kappa beta one side for inflammatory reaction. Other side, they can also get involved in insulin pathway to create insulin resistance and increase the adipogenicity or deposition of fat. And this mechanism is now becoming obvious and more diverse. So now we know that the obesity is not just linked with the genetic makeup, it is more to do with the microbiota. So thanks to metagenomics, again, that the microbiotas are important for our obesity. And you'll be also interested in a very good experimental experiments on them in animals as well as in human. Say what they did was the gut microflora, we call it fecal microbiota, when they transplanted from the obese animal to lean animal, or obese individuals to lean individuals, lean individuals tend to become fatter. With the same food habit, they're becoming fatty and becoming obese. And vice versa, were also true when the lean individual gut microflora or the fecal microbiota was transplanted into obese people. Of course, this was less successful, but these were more successful. Such experiments had been done in animals, done in humans, and they say that now microbiota is more important. So beyond going this, now we have the technology of the metagenomics, which is discovering new pathways, new mechanisms, new methods by which we can diagnose disease, we can find a new methods of prevention, and we can divulge new insight into diseases such as metabolic syndromes, obesity, and whatnot. I need not tell you the importance of microorganisms in skin, in milk, in other, even now we are talking about microorganisms being present in brain, what we call as brain microbiota. I'll come back to later. So now we are in a, we all know that microorganisms are present everywhere. They are present in skin, brain, liver, they're present in blood. The tissues which were once thought to be the sterile, like brain, which were thought that there is no microorganisms, even now we know that there could be the presence of microorganisms there. So microorganisms are everywhere. They play a very important role. The total number of cells that we have in our body is about 30 trillion. 30 trillion, which is largely, about 25 trillion comes from the blood cells alone. Whereas the number of microorganisms present in the gut alone is 38 trillion cells. Leave alone the numbers that are present on the skin, the numbers that are present in the other organs, in milk or other places. So number of microbiota that are present in our organism, in our body, is far higher than the actual number of cells that we have in our body itself, the body cells itself. So important the microorganisms have been. And the era of metagenomic is something which is diverging these informations, and that's where it comes into the role. Now we are trying to profile microorganisms in every organ such that we can know what are the normal microbiota of an organ in a healthy status, in a diseased status, in a different physiological conditions, different, let's say, lactation period, different feed habits, different dietary schedules, and so on. Microbiome profile has now become a hotbed for many of the things, and many new discoveries are coming up which were hitherto unknown to us. Now let's say the question, what is metagenomics? I've been saying metagenomics, what is metagenomics? So when you say the metagenomics, we have two words, meta and genomics. So it's genomics of all the microorganisms that are present in a particular habitat at a particular time point. Say, for example, all the algae, virus, bacteria, fungi, proteases, protozoans that are present in rumen of the cattle can form the rumen metabiome, and analysis of that, the genome of all of them, is what constitutes the metagenomics. Now, how to do the metagenomic analysis? How to analyze, how to measure and interpret the metagenome of all metagenome of a particular tissue at a particular time point in a single go, how to analyze them. Traditionally, in the past, we have been doing this with the methodologies of culturing. Earlier days, we all know it, we used to culture one or few, say, for example, if we have the wound sample, the pus, we can take that, culture them, find out few organisms that grow. Many of the organisms will not grow depending on your culture conditions that you're providing. Say, for example, if you're growing in a nutrient agar, or if a nutrient, or in a blood agar, or in aerobic condition, anaerobic conditions, depending on that, the number of organisms will grow. So that was not possible. Many of the organisms cannot be identified with that. Initial days, a variant of this method, of traditional method, was to do what we call as culturome. So we can take the microorganisms, group of microorganisms from our desired sample, dilute them in such a low dilution such that you have one microorganism or one bacteria, few microliter, and distribute them into 96 well plates or 384 well plates, such that in each well plate you have one or two bacteria, and then grow them so that each will have one colony. That colony will be representing of a particular bacteria. We can take them and go for the Sanger sequencing and know what are the microorganisms who are present. But again, such traditional ways of knowing the microorganisms present in sample have been culture dependent, and we fail to identify many of the organisms for which the cultures were not done. Say, for example, if you take milk samples, you may be able to get hardly E. coli or maybe Staph aureus in a healthy milk sample. You will not get beyond that. But now we have newer technologies, largely based on the next generation sequencing and the third generation sequencing, which in the previous lecture you have heard of. It is possible now that we can collect the DNA sequences from complete microorganism population that are present in a particular sample and directly sequence them, map them into genome, and find out what are the all organisms present within them. So there is no necessity of culturing the techniques. Directly take them, sequence them, and we can find out what organisms are present. We can do the finding of what organisms are present. We can even go beyond that. We can find out what are the characteristics, what could be the function in that particular habitat, say wound or diabetic wound, a normal wound, or a normal gut microbiota, diseased gut microbiota, normal healthy milk, the mastitic milk, and so on. So the metagenomic is such a important and high throughput tool, such a valuable tool, that we can do that all is in a single go. So how to do it? So there broadly, what we use currently is two methods we use. One is what we call as Amplicon sequencing. In Amplicon sequencing, say I'm talking about, say for example, bacteria. What we do, we take certain DNA sequences which could be used as a proxy to that particular the organism or that bacteria. In case of bacteria, 16S rRNA is something which is common. So DNA encoding the 16S RNA is amplified by PCR. So particularly the hypervariable region V1 to V9 is something which is the common target, and there are variations in the sequences by which you can identify the different species. So we can amplify, say for example, V3, V4 regions, and this amplification of these genes can then be used for mapping and finding out what genome is present. So how we do it, typically, say if you have the if you have oral sample, you want to know about the microbiota of your buccal cavity or the fecal matter, you can collect the sample, then isolate the DNA. After isolating DNA, you can amplify the 16S, the DNA for the 16S rRNA, which we call it 16S rRNA. Now each one has their own challenges, which I'll come back to that later. After amplifying these sequences, you can use normal PCR to amplify. Usually two-stage PCR is done. First stage we amplify them, and next stage we can attach certain adapters so that these sequences can be used for sequencing. And if we want to have multiple samples to be sequenced together, it is also possible, because many of the high throughput machines will allow that. Say for example, the Illumina sequencer or the nanopore sequencer or PacBio, even Pyro sequencer will allow us to do what we call as barcoding. we can, each sample, say if you have 5 samples, we can take 4 samples, we can take these 4 samples. In each one of them, in the sequences, we attach a specific sequence which is unique to a particular sequence. We call that sequence as barcode. Then we sequence all of these sequences and go for the analysis, and after analysis we can demultiplex them to find out what these individuals are. So how to do the analysis? So if you want to do the Amplicon sequencing, take a sample, say gut sample, means take a gut sample, isolate DNA, do a PCR amplification for rDNA. So primer pairs could be, you can use those primer pairs which span, which have greater coverage on a wide range of organisms. Many companies will provide now ready-made primers, or you can go many published report from where you can take the primers, or you can design your own primers in certain databases, say for example Silva database or the ribosomal DNA database, where you can map and see your sequences is mapping to many. You can do ePCR to find out, and then using that primer you amplify your gene, do barcoding and the adapters, and go for the sequencing. How to do sequencing, you have already talked about it in the previous lectures. So you do the sequences and you can generate the data. Now what we do with the data, say I've taken the example of here Illumina sequencing, which is very, very common. We also follow Illumina sequencing. Of course, we have now moving on to nanopore, which is a large read sequencing method. So typical sequencing data will come in a FASTQ format, which is basically, as you all probably know, is DNA sequences along with the Fred quality score. So you can determine whether they are good or bad. What is the chances of being rightly identified? So usually Q30 score is usually taken. Fred score of Q30 usually taken for this. So you get a FASTQ files. If you are doing in your company, in your laboratory, you can do a base calling and generate a FASTQ file by the machine. If you are doing the company can directly provide your FASTQ files in the CDs or through a FTP site. Once you do this, get the sequences, you have to do demultiplexing. Demultiplexing, which is basically, depending on the barcode, you can separate out the data sets for each sample. And these sequences are usually done what we call as paired end reads. You would have talked about this in the previous lecture. Both the DNA sequences are sequenced, both the sense strand as well as the anti-sense strand. So you merge the reads from the sense and anti-sense strands and trim out the barcodes, trim out the primer sequences, and do certain quality control to discard data sets which has poor coverage or poor the quality score. So for that we have several softwares are there, but which can be used, say FastQ or FastP softwares can be used. Trigonometric is another one which can be used. So they will generate a FASTA file. You can get FastQ also, but generally we generate them as a FASTA files, which is basically DNA sequences. It will be multi-FASTA format for all the sequences that have been sequenced. These sequences can then be mapped to databases to generate what we call what organism they represent, from which organism these sequences has come, which species or which phyla these sequences has come. So there's two methods of doing it. One method which has been used earlier and widely is what is called as clustering. Each sequences are clustered. Say if they have a similarity of 97 and 98%, we cluster them together and map them to a database, say Silva database, Green Genes database, or ribosomal DNA databases, to find out which organism they build. So what they generate is OTUs, operational taxonomic units, which is believed that each sequence is representative of one particular sequences. Now this method has a limitation that in databases may be incomplete, and certain variants or homologs of the 16S rDNA sequences may not be present. So relatively newer strategy is to use what we call as generate the ASV, Amplicon sequence variances, and we have nice softwares to do that. there is tools available within software. The most common tool that we use is QIAM. Now QIAM 2 is available. They have denoising, or QIAM has deblurring option. There's another one, DADA. DADA 2 will have denoising options. You can also use usearch. Usearch can also be, it's had many tools combined, bundled together. So you can use these tools to generate ASP. So these OTUs and ASVs will basically generate a taxonomic table. Means in each sample, what organisms are present and what are the read numbers on that. Based on these, usually our analysis ends here from the sequences. But it is also possible that you can use these sequences to predict their functional tables. Means we can go for gene ontology and pathway analysis. We have software such as PicRes2, which can map your sequences to databases or gene ontology databases or cake pathways to know these organisms are involved in, say, nitrogen fixation in case of choice samples, or it's some diseased. You can map it to big bug-based databases, which which is the database of the medically relevant organisms, what diseases they are linked to, say Mycobacterium tuberculosis or some other diseases. So those things are possible, but this has own limitations, not commonly used, although they are doable. They are less reliable, less robust compared to what I'll talk later, the whole genome sequencing. Now, once you get the taxonomic table, from the taxonomic table, which will have the organism that are present and the reads, you can do many things. You can do many statistical analysis to calculate, say, for example, the alpha diversity, which is richness and evenness of the organism in a particular sample. Means how many types of organisms are present, and what are the individual organisms richness within them. That's what richness and evenness. Usually we do that by doing what called Shannon's index, which ranges from 0 to 1. We can get an idea about the alpha diversity. We also can create a rarefication curve, which will give you again an idea about the alpha diversity. We can also calculate beta diversity, which will give an idea about the diversity of organism between 2 or more samples. So we have indexes for that, Berry-Curtis indexes, we have impact indexes, we have, and so on. So we can make different types of tables. We can also do molecular phylogenetic analysis, say different software such as Mega and all we can use to find out what organisms are present and how they are interlinked over the period of evolution. We can generate corona chart and all to show the interrelationship and all. So Amplicon sequencing basically gives this much of idea only: what organisms are present, what is the richness, what is the diversity. And Amplicon sequencing is largely done for the bacteria, archaea. You can do it, say for example, eukaryotes, you can use 18S RNA sequencing, the ITS sequences you can use this. The algae you can use the 23 RNA sequencing and all, but it cannot be done for viruses and all. It's not possible to do it. So those are the limitations of the Amplicon sequencing. There are many softwares available. As I said, there is one famous software is Mothu. Now more commonly we use QIAM. QIAM 2 versions is available. DADA 2 is also available, which is mainly for the Illumina platforms and to generate the Amplicon sequence variants. uSearch is a relatively newer one. It's very fast. See, all these sequences analysis requires computing power and time to handle the data. uSearch is relatively new. It gives much faster. Among these are the common tools, but there are many other tools, and these tools are usually built up with all the other software that are required for analysis of sequences, as I mentioned earlier. You can even generate data on the alpha diversity or the beta diversity, rarefication curves. All this can be generated using these softwares. This software generally works on Linux, but there are versions online like Galaxy where you can do it in the web servers, can also be done. The databases that we use for analysis of 16S RNA sequences, the most common used ones are Silva, which contains the database for both 16S as well as the 18S RNA. So good if you are suspecting that your samples will have contamination from eukaryotic and all, or you can pin both will be there, so it will help in filtering. Green genes and ribosome. Once upon a time, this used to be very, very famous, but these databases differ in the number of entries within them they have. Some softwares will not only match your DNA sequences with DNA, they also use protein mapping and all, the codon mapping, and also other databases are also used like KEGG for the pathway analysis. You have seed to make the subsections, and then we have PFAM, protein family, and all. So among these, Silva, green genes, and ribosomal databases are commonly used. Certain softwares will, when you analyze downstream, will be specific to certain software, certain databases, so that you have to deal with it. And it is not necessary that you have to use the 16S rRNA alone to do this. It has its own limitations. Say, there could be horizontal gene transfer of rDNA, which will confound the reliability of the data sets. And 16S rRNA also exists in multiple copies. Earlier, it was approximated that they are in approximately 4.1 copies per genome, but now we know that it can go as much as 15 genomes. So that's one of the limitations. So people are looking for alternatives to 16S rRNA or sequences, some of which is very prominent candidates are RPOB, others are AMO, PMO, the PUFM, and so on. These are the sequences which people are exploring. Housekeeping genes, the advantage is that the horizontal transfer of these genes are less likely. That's the basic advantage. The disadvantage is that they are still insufficient for inaccuracy. So what basically do is that we go for the combine this with the 16S RNA sequencing only when we find there is a limitation. So if there is a limitation of 16S RNA, combine that with the housekeeping gene and then try to analyze the data. Now people are also considering using multi-locus sequence typing. So that's also emerging up, new one. But as of now, the most commonly used one is 16S rRNA for bacteria, 18S for eukaryotes, and ITS also, and 23 for algae. Now, as we said, the amplicon sequencing is possible only with bacteria, archaea, not possible with viruses and majority of the fungi. And they are also suffering from the amplicon bias during PCR amplification. Your PCR, depending on the primer bias, depending on the setup, the amplification or the thermal profile of the PCR, or the specificity of the primer itself, you can have biases. This is the disadvantage of this, in which case the other choice is to do the whole genome sequencing. Nevertheless, amplicon sequencing has its own benefits, its own advantage. It's much cheaper. Whole genome sequencing, as we'll talk very shortly, can be used to profile the complete microbiome, including viruses and fungi. And there is no amplification bias here in this case. Of course, a PCR is involved, but that is PCR is for making library, not to amplify the gene as such in the before sequencing. So the amplification bias is not there. But the disadvantage of the whole genome sequencing is that it's expensive. It requires a lot of the computational power. And as we'll see shortly, the computational programs that are available, they have their own limitations. They are far from complete. That does not may not give you the correct representation. Sometimes there might be error. On the other hand, amplicon sequencing is very, very cheap, easy to do it for large sample. See, even if you are outsourcing it, I think you can do it somewhere around 10,000 to 12,000 rupees per the sequencing. You may get it done. Another limitation of this is the resolution of amplicon sequencing is less. You can get at the best the genus level. If you sequence the whole genome, then we can get it the sequence is higher resolution up to the species. It might be possible, and as I said, you can even go beyond to find out the genes and pathways. So how the whole genome sequencing is done and what it is. So in whole genome sequencing, what we do is basically we take the whole genome of organism, we collect the samples, take the whole genome, and fragment them into small, say by sonication or something, we can fragment them into smaller fragments, and each of the sequences can then be sequenced by massive parallel sequencing such as Illumina or PacBio or the nanopore sequencers. So this is the outline of how Illumina sequencing is done. We collect the sample, suppose whatever you want to collect, the swab, oral swab or gut microbiota or milk sample or whatsoever. You collect the sample, you isolate the DNA, fragment them into smaller pieces by sonication or other methods, and then prepare the library. Now this library, you may, those who have attended the last lecture, they know we do what we call as the bridge PCR. Illumina implements bridge PCR so that each of these fragments can be developed into multiple copies, and in the all of the fragments may be represented. So what we call as the library of the DNA. We then do a series of processes. We repair the ends, add overhands, and load them into the flow cells, and then do the sequencing. So at the end of this, when you do the sequencing, again you get data that can be analyzed here. So the data that you will get here again will be in the FASTQ formats. Okay, FASTQ formats, as I said, will have your sequences, nucleotide sequences, along with that for that quality scores based on which you can determine whether it's to be accepted or not accepted. So when you have these sequences, first you have to pre-process them. You have to remove the adapter sequences that are usually added during the library preparation for bridge PCR. And you can also remove the other sequences which are of low quality, low Fred score. As I said, usually EU30 and all is preferred. And you may also like to do filtering. See, if you are collecting, say, blood samples, and you are targeting only for the microbiota, it will also have, even if whatever purifications you do, you will have some contamination from the blood samples itself. So you would like to filter them out. So you can use program. There's a number of software we can use. Trigonometric for quality control. Bauti can be used for filtering. Or there is another pipeline called need data, which is incorporating all of this to do the filtering as well as the processing. And this filtering is very, very, very vital, as we'll see when we are talking about the challenges to this. When you collect the samples, say, nowadays a lot of talk is going on about brain microbiota. People say there's some report, they said brain, which has very solid, strong blood-brain barrier, doesn't have microorganisms unless they're diseased when the brain blood-brain barrier is breached. But then people have found that there could be microbiotas are involved. And that microbiota, they say, is naturally present, and that may be the cause of the neuronal disorders or neurodegenerative diseases that may be related to the health status or mental status of the animal, stress conditions of the animal. So a lot of research is going on. But then there is this is a controversial. Some say that this might come because when you collect the blood samples, brain samples, there could be some blood may have gone while dissecting it out, and blood also contains approximately 1 to 10 million bacteria per ml of blood. So it could be that. Some says that, even if you do the kit, kit themselves may have some microorganisms. So you are obviously going to get some sequences from them. So it's very, very vital that you do the filtering and sample processing. So need data is very, very vital here to do that. So after doing this processing, what you will get is the FASTA files. Now, FASTA files are then mapped to be the genome into this. We have two approaches to do that. One is called read alignment to curated database. Other one is called assembly-based methods. Read alignment is, see, the small sequence that we get is called as reads, as you all know it. And we have a databases of all the organisms, say NCBI databases, which all of you are familiar. So we can map these reads into those databases and then find out which set of the organisms they belong to which organism. and we can construct the taxonomic table. To do that taxonomic profiling, we have very commonly used software is called Kraken 2, but we also have other programs like Metafile N. Second version is available. Kraken 2, Kraken HTL is also available. There are so many programs. I'll give you a list very shortly. And then this one, again, as I said, just like 16 sRNA, we have number of programs, say PicCrust can be used, or Tax for Fun. Tax for Fun is an R package. You can use it to generate data about the taxonomy to function. If you know the organism, what functions they may have, say environmental function or metabolic function, nitrogen fixation, iron metabolism, or disease causation, even we can map to, as I said, bug base, for example, then we know whether they are gram positive, gram negative, or they are linked to certain diseases and all. So those information can be obtained. PicCrust is usually attached with the green genes databases, and this can give you about gene ontology with the KIC pathways, and also many such programs are there. FabProtex is another program which can be done to find out the environmental information on the bug base, FabProtex, and so on. So this can be done in this. The other way of doing it, this read database can also be used to do functional profiling. What functions they associate? We have programs, very good programs called such as Megan. For human, we use human N2 can be used, which can generate the gene ontology and all. So either we can go this way or this way. The other way of doing it, particularly if you're expecting certain sequences which are not likely to available in the gene database, or if you are trying to create the draft genome of certain microorganism, you may choose to go for this pathway, assembly-based pathway, wherein we take the reads, assemble them one by one to find out the longer sequences. So what we do is here we have read sequence, we assemble the reads by programs such as Spades, MetaSpades, MegaHit. The number of programs are there, each having its own advantage and disadvantages. Each has different algorithms, and that's in fact is a challenge. But the way it exists today is that we have to use one or more of such programs and assemble the reads and develop the contigs. And these contigs, which are long reads, what you get is now is several reads assembled together to get long contigs, longer sequences. And then we try to predict whether these sequences are a gene or a non-gene. So you may have studied about in bioinformatics classes and gene prediction tools such as the GenMark. Metagenomics is one program which can be used for metagenomics. Proca is another one. And then you will get number of a list. There might be certain sequences belonging to more than one sequence. So you can remove the duplicates. We have several programs such as, for example, CD-HIT, which can be used to remove the duplicates and create a gene list. Now, gene list can then be quantified based on the read count and all, to using programs such as Bauti2 or Salmon. So what you get basically end result is a gene abundance table, which is gene list along with the relative abundance of the gene in the sample. That you can get it. If you, as I said, if you are going to do the assembly of a draft genome, or if you are going to find out the gene clustering to different sets, so we can use other programs such as the MetaRap or the MaxBin. This is basically binning of contigs into a particular gene cluster, and then we can generate the functional tables from this. So these are the two approaches we have: read-based and assembly-based. Assembly-based methods are more computationally more demanding, takes the greater computational power, and usually useful when the sequences you find that are unknown, or if you're making a draft genome or something. See, compared to Amplicon sequencing, whole genome sequencing requires a lot of computational power, while 16S RNA database or Amplicon sequencing data you can analyze using your laptop or your desktop. It is usually possible. You will not be able to do so in whole genome sequencing. Whole genome sequences will give databases in gigabytes or sometimes even terabytes. So if you're doing core samples such as the dung sample or the oral swab or the fecal sample, which are uniform population, you may get up to 5 to 10 GB of databases, sequences. sorry. But if you're using the complex or mixed samples, you may have 30 to 300 GB of databases, for which you require parallel processing, managing of the data. So usually most softwares for these are available in Linux. Many R packages are also available. You may require to, if you want to handle them properly, Conda, BioConda are usually commonly used to handle this, and the GNU paralleling and all, you need to do that to assign the task, run multiple things in parallel. So this is computationally challenging. Software, as we'll talk later, has a lot of issues. So I have given here the list of various softwares that are available for QC control. In the beginning, I said if you are doing pre-processing, you have number of them. Fast QC is very commonly used. Trigonometric is another very commonly used. Although there are other fast QBB tools, QAdapt, each one is considered equal. Trigonometric gives a lot of flexibility, very common, and you can do it even in the web servers such as Galaxy, but again, it takes time, and sometimes whole genome sequencing data is difficult to do in Galaxy servers. For classifying or aligning, you have, as I said, Kraken is one very common, but we have Kraken LL. These are basically k-mer-based algorithms, usually tetramers and their frequency and jet scores, they do it. Your Clark, Telescope, Clam, GADVASU, Diamond, and so on. We also have Blast Plus. We have FTP server. We can get the whole database into your system, and then you can do it locally in Linux environment. You can do that. Gotcha is another, centrifuge, and so on. There's a big number of tools. Now you may get into trouble which tools to be used. Each tool has its own advantage, disadvantage. There are a number of articles on this which are comparing these articles and say that which one has limitations. Also, usually people combine. The most common one people use is Kraken. Is very, very common. If you are doing the assembly, so we have megaHit, spades, megaspades, Raymeta, Velvet, MetaVelvet, Velvet, and so on. These are the common programs that we have. Similarly, you have for assembly the contig binning. If you are doing for binning, we have MaxBin. In fact, MaxBin 2 is now available, which is very popular. MaxBin, you have Concat, MetaBat, and so on. You have MetaQuest for assembly and binning in the quality assessment on this one. So MaxBin 2 is one of the very common used ones for doing this. So number of softwares are available to do this. These softwares has been used one after the another, and so you have to, the pipeline I have already described, so that pipelines are usually done in the Linux environment. So before I go into applications of it, let's quickly give a recap of what we have talked so far. We said about amplicon sequencing and whole genome sequencing. Amplicon sequencing is usually for identification of organisms and their diversity analysis. Usually it doesn't go beyond it. If you have a large number of samples, or you just want to know what organisms are present and what is the richness, then what is diversity, alpha diversity, beta diversity, Amplicon sequence is cheaper, easy to perform, easy to do it. Some of the limitations of this is, he said you cannot do it as viruses and all. If the sequence is not known in the database, you cannot do that. So sometimes if you get sort rates, difficult to identify unambiguously, but that's something we have to deal with everywhere. Whole genome sequencing, on the other hand, gives a complete picture of genome, including the identification of species, to identifying different genes present therein, the location of the genes, different operons present then, the transcriptional units, control elements, including translational units that are present within them. So this gives the resolution that you get with species is you can go to species level. Here usually we can go up to genus level, not beyond that, because the 16S RNA sequences will not have that much resolution. Here you may get, if you're lucky, and not lucky is usually you can get up to the species level, so greater resolution. You can get phyla, order, class, all genus, species, all the levels you can get the resolutions and find out. So you get a wider range more detailed description. And as I said, you have softwares to get better idea about different genes. We have another variation of metagenomics called as metatranscriptomics, which is very similar to metagenome sequencing, but the starting material is RNAs, and that can give you an idea. So doing an RNA sequencing, you get to know about what genes are functional in a particular time point in an organism or in a group of organisms. The limitations of this whole genome sequencing that you should be aware is that one is it's costly and time consuming. While you can get 10 to 12,000 rupees if you outsource Amplicon sequencing, 10 to 12,000, but whole genome sequencing may 40 to 50,000. Of course, if you're doing your own laboratory, it might be cheaper, but cheaper if you're doing in large bulk volumes. And this one requires a complex bioinformatic algorithms and softwares, many of which you can get it for free. They're open source, but the pipelines are such that you need to have some expertise in handling them. So a use of a statistician or a computer biologist or a bioinformatician, to be more specific, is very, very vital for this kind of analysis. And we all, as I talked earlier, each software in the pipeline that we use has different algorithms, and each algorithm may give a different result. So that again is a challenge. And it may not identify all elements, particularly the longer fragments, large operons, something which is having say 5 kb and all. Longer sequences is very difficult to identify even with whole genome sequencing, because you get small pairs. So now let's see, we'll talk about some of the applications of metagenomics, where we can apply the metagenome, where we can do this, how we can use it in our research or product development, or how the entrepreneurs can use this technology to develop newer product or a newer process. One that we have talked is we can use this to screen a complex microbiota in a clinical environment or in a environmental sample. Say you have pus samples from diabetic, research has shown that if you take a wound sample from the diabetic ulcer and non-diabetic ulcer, they have a different population of cells. So that gives an idea that what kind of microbiota are there and how many they cause. Some of them are useful. Now you'll be surprised to know all the bacteria in the wound are not harmful. Generally, we believe the bacteria in the wounds is going to cause the necrotic effect, going to cause some or the damaging effect on the cell. But metagenomes have revealed that some of the bacteria may help in the proliferation of healthy cells. Some of the bacteria may help in bringing the inflammatory cells by chemotaxis so that the healing could be faster. And particularly in diabetic wound, the population is very, very different. So you can get this idea about these microorganisms, which may help in not only identifying, screening the organism, but also finding out which organisms are causing the problem, and you can choose appropriate therapeutic strategy, antibiotics or the antimicrobial peptides for treatment of those drugs. You can also do that in environmental samples. Say you have water samples from arsenic. Arsenic is a very big problem in certain parts of our country, like Bihar and West Bengal. So many areas are predominant with arsenic organisms, and you would like to know that you can expect the organisms or bacteria which are thriving on arsenic rich water sample or soil sample. That means they are capable of metabolizing them or sustaining them. So some of them might be useful for devising new strategy for arsenic remediation. You can take the samples, the water samples or soil samples from arsenic rich area and a clean area, and then combine them to find out which organisms are predominant and how they can be used for the arsenic remediation. Or say if you take it for petrol bunk, and you can find out what can be used for phenolic removal of phenolic oil, or newer bacteria for dealing with the microplastics, which is becoming a headache nowadays, or oil spillage in seas, all those is possible. You can do, of course, phylogenetic analysis. If you have the grid samples from a particular source, and you can do phylogenetic analysis, that's very, very basic that we can do it. The best thing is, which was hitherto not possible with cultured organism, was the catalog of organisms that we know in a particular habitat was smaller. With the metagenomics, now we are getting to know new organisms which were earlier not known because they could not be cultured. So new uncultivated organisms are being identified. The catalog of bacteria are increasing. As you will see shortly, say for example, as I said, milk. If people, when you culture the animal milk or human milk, we usually get one or few organisms in your culture, say E. coli, non-pathogenic E. coli in case of healthy milk, or Staph aureus and so on. But when you did the metagenomic analysis, it was revealed that there is a very large population of organisms that are present. So is the case with blood. We think that blood is sterile, but no, blood is not sterile. Metagenomic studies have revealed that there are up to 1 to 10 millions of bacteria per milliliter of blood. That's what was not possible earlier. So you can identify new uncultivated organisms, those we could not identify with culture only. We can also use to understand the different populations within a microbiota. Say on the skin sample, skin, we know that many bacteria are residing. Some of them we know, many of them we don't know. So under different conditions, dry skin versus the moist skin, or the healthy animal versus unhealthy animal, what are the organisms of microbiota that are present, and how they interact with themselves, how they interact with the environment. That analysis can be done with the metagenomics. Say in the skin, for example, we know that certain microorganisms releases antibiotics or antimicrobial peptides which kills other organisms, which are an important source of discovering new antibiotics which were not known earlier. That's where I'll talk more about this later. And we can also use this to identify novel enzymes of industrial importance. Say, it has been done that xylase is an enzyme which is very useful for pulp industries. Cattle rumen have microorganisms which can digest xylase, the plant tissues by xylases, and these xylases has a lot of application in there. So the certain scientist has explored the metabiome of the rumen and then found that certain organism has xylase productions which in a good amount that could be increased and can be fermented in a large fermenters to produce very stable xylases for industry application in pulp industries. So novel enzymes, novel antibiotics can you find out. Probiotics, the functional foods, we know many of you are available. We talked in the beginning of this lecture how we can use this for, say, the obesity or the metabolic syndrome, treatment of metabolic syndromes, or treatment of mastitis. See, if there is a microbiota, say for example in the milk samples, if there is a microbiota is available, they are in a particular state of homeostasis. If there is a dysbiosis, if the microorganisms are imbalanced, then obviously the metabolome of that area will also be imbalanced, and vice versa. So changes in the metabolite composition can have effect on the microbial composition. So it might lead to development of new metabolic modifiers or probiotics that might lead to better treatment of the diseases. And I'll give you a very good example of this probiotic and metabolic modifier shortly, taking some examples. Let us take some examples of this. So milk microbiota, an area where we are working now. Earlier, it was believed that milk microbiota is milk is traditionally considered there's not much microorganisms there. Milk is considered usually sterile, but there are a lot of microorganisms. We knew it. with the advent of next generation sequencing, particularly after the pyro sequencing and Illumina sequencing, and now more recently the nanopore and PacBio, we found that the animal milk as well as the human milk has a plethora of microorganisms which were earlier unknown to us. Although we do not know what is the source of it, the metagenomics also revealed, it's a comparative analysis and lineage tracking using statistical tools has revealed that the intestinal metabiome or the milk microbiomes are very much related. Of course, this study is still going on. They say that in the intestine, the metabolic analysis have revealed, and fetal maternal transplantation studies, these two studies combined together, has revealed that the gut microbiome may go into the epithelium, may go even into the blood, may go even into, may be taken up by the microorganisms, the macrophages and all, travel all the way and come into the milk, the udder. Not just the milk udder, they may go onto the liver. There are documented evidences that ruminal microorganism can pass through the ruminal epithelium, go into the blood vessels, lymphatic vessels, and cause the liver abscess. There's the documented evidence, thanks to metagenomics. Similarly, there are evidences that say that gut epithelium may be crossed by the microorganisms that are, or the microbes that are present in the epithelium, go from the mother's gut to the lymph nodes and the blood circulation, from where they may come into mammary gland, and from the mammary gland they may be poured into the milk microbiota. And this milk microbiota is important for the homeostasis. We may ask question, why? See, nature, as we all know, if there is something that must have a purpose, so the question comes, why does the milk microbiota has? Does it have a function? So that's another. We believe that it is because of the symbiotic relationship that they may have. Just like skin, we have symbiotic relationship. Milk microbiota probably also has a symbiotic relationship. And this microbiota is very, very important, not only for the animals themselves. We'll talk later shortly that get the milk microbiota can go back into the other area of the body. And we all know that this microorganism can go into the infant. So the milk microbiota are also important for colonization of intestinal or gut microbiota in the infant, in the baby, and this applies both for the humans as well as the animals. So how important it is. Milk is not just a source of nutrient, not just a source of the antibodies in the early stage of life, but also they are vital for establishing the gut microbiota. And we'll also shortly, the gut microbiota in turn are linked to the health status of the animal. They are linked to the feed intake. They are linked to the growth rate of the organism. Depending on the different types of microbiota can cause different rate of growth, different rate of the feed intake, and that in turn will affect overall health of the infant, the growth of the infant, and that has direct relevance to the animal's productivity. And that has also direct relevance to the health of the consumer, human, who consumes the milk. Same milk, when they come to the human, they will also become a part and parcel of our gut and will affect the gut microbiota. Dietary influences, we all know about it. And then in human, we have, in the beginning, because they could have the obesities and the being lean are linked to it. It is linked to the several metabolic syndromes, linked to the stress even. I'll talk about that shortly. So number of studies has been done and number of organisms have been identified. This study is ongoing. There in many species. Say for example, you see cattle. There are different research reports dominance of different species. There is no commonality. This may be because of dietary variations from the animal to animal. This could be because of the regional differences. Animals of Indian origin, animals of Malaysian origin, animals of that origin, US origin have different feeding schedule. Say, for example, if you feed concentrates, they may have different the microbiota profile. This could be different parity level. This could be a different level of lactation. So there is a lot of variations between the cows to cow. And as I said, these sequences nevertheless have increased the number of organisms, and there's a breed differences. In a very interesting study conducted in India in Anand Agriculture University, the three very nice Indian breeds, the two Indian breeds, sorry, the Kangaraj and Gir, they compared the milk microbiota and compared them with the crossbred animal. So when they did the culturome, when they cultured the organism, they found that Kangaraj had only Escherichia coli, Gir has E. coli, the crossbred has Staph aureus. Now where from it comes remains to be discovered. When they did the metagenomic analysis, they found that the culture-dependent identifications have missed out a lot many other organisms. See, in case of this, only 30% were E. coli, rest all belong to many other species. In case of Gir, only 26%. In case of cross with Staph aureus, they present less than 50% of the organism. There were many other organisms which were not found. Even phages were found, which were fast bacteria, viruses were found, which were not known to be present in the milk. And when they tried to see between these Kangaraj, Gir, and cross, but there's distinct differences. Say, in case of the Kangaraj and Gir, there were dominance of proteobacteria, whereas in case of crossbred, Firmicutes. Firmicutes are more linked to obesity. They are more linked to greater feed conversion ratio. So it could be, we do not know the reason, but could be because crossbreds are generally bred for the purpose of greater milk production. They are fed high concentrate rich diet, which again changes. There are studies which says that the concentrate feed diets can increase the Firmicutes and so on. If you look at the family level classification, there is the Enterobacteriaceae family were seen in almost all the cases, whereas other species were like Bacillacea were species-specific differences. In the Enterococcaceae, Streptococcaceae, there were specific differences within these organisms. So there were species-specific differences, and number of studies are there. Metagenomics says that the milk microbiota can vary with that. Even we have the data for this. We do not have much data for many of these species, say goats, which are so important for our country, sheep, and the native animals. Many of the microbiotas are not available to us. It's an area of research. Back in our lab, we actually try to do analysis of microbial population from different databases from different countries and different animals. Try to see, was there a common organisms present in all the samples in different countries. We took African countries, US, Canadian, Chinese, and Indian population, and we found very interesting thing. Certain organisms were present in all the cases without exception. Certain were present in 4 out of the 5 populations. They were present. Some were present in 60% of cases and all. So it suggests probably some of these organisms may evolve as a microbial signature of a healthy population. And then if you know the healthy population, if we can, there are simple ways of doing this microbial population analysis as we talked about. Say, if you use the 16S RNA sequencing, nanopore sequencer gives a very simple kit, and it could be done at a lab level itself, at the farm level itself. So if you can find out the microbial signature of the healthy, then we can use it to diagnose the unhealthy cases. Particularly, mastitis, subclinical mastitis, which is a very big problem in our country. Country is trying to double the farmers' income, but mastitis, particularly in subclinical mastitis, where the symptoms are not overt, the microbial profiling may probably come out to be a diagnostic marker, and research are ongoing into that. And if we can find out, we can probably also look into the metabolome and see if some metabolic modifiers can be used to alter this, the microbial profile of unhealthy animal back to the healthy animals. Or it may also be possible to develop certain probiotics or prebiotics which can be kept the animal's health into healthy status. So we take probiotics for gut microbiota. So far we haven't thought of the probiotics for the milk microbiota. So that could be a future therapeutic strategy which only metagenomics can help us to find out, and areas of it useless. There's a very interesting study. I took it here to understand how important metagenomics could be. So this is a metabolic profiling of healthy and mastitic milk, sorry, intestine. So what they found that if you have mastitis animal or healthy animal, obviously milk microbiota will differ. Not many works are there. This study in 2018, they found that in healthy and mastitic cows, the intestinal microbiota will be also different. So the animals which chose high somatic cell count, they selected those animals, performed the 16S rRNA sequencing as well as the whole genome sequencing from the healthy animals and the mastitic animal, the intestinal microbiota, and then found that there are clear differences in the microbial population. You can see certain populations were very, separate. The PCO analysis, principal component analysis, the predominant in mastitis, and so were the case with the whole genome sequencing. So certain organisms, in mastitic animals, the microbiota were disbalanced in the intestine. The other report which says that even the endometrial microbiota may also get changed. That report is also there. Now, what these guys did is very important, very, very interesting. They took the fecal microbiota they collected from the healthy animal, and they also collected the fecal microbiota from mastitic animal and fed it to a healthy mouse. So when they fed the healthy mouse, so it is quite expected that in the animal, the transplanted the mice, okay, it was mice. When they fed the fecal microbiota into them, fecal microbiota from the mastitis cows, the intestinal microbiome of animals got altered. There was a dysbiosis, and they tried to be similar to those of the mastitis. So you can see they have changed the metabolic profile of the animal. Now, more interesting thing is, in these mice which received the fecal microbiota transplantation, after about 21 days, these mice developed mastitis. That means milk microbiota for mastitic animal, when consumed by the consumer, may lead to the other infection. Here it's not just mastitis, the animal they also develop, there was inflammation in other organs like liver, inflammations in the spleen, inflammation in jejunum, colon, many places they found. Even spleen had the inflammatory symptoms. So the transplantation of gut microbiota from the mastitic animal led to mastitis, apart from other diseases. That raises a concern, and that says that how the one side is health impact. Second impact is that we may also see how much impact it may have for the human health, and it also gives another suggestion, another association is that the the gut microbiota are involved in causes of mastitis. They may not be the full cause, there may be other causes as well, but gut microbiota is also involved in causes of mastitis. So feed habit or anything that influences the gut microbiota can lead to other inflammations as well. So that now comes to the gut microbiota. Now we know that gut microbiota in further studies. Now these studies have shown, if you look at the gut microbiota, within different segments of the gut, you find different aerobic status, different pH level, and we have specific population of microorganisms. Studies have shown say for example stomach, which has a much low pH 1 to 3, there are microorganisms such as predominance of lactobacillus, streptococcus, staphylococcus, enterobacteriaceae, they are predominant. And in duodenum, if you see, or jejunum, the numbers increases. We all know this in jejunum, ileum, cecum, we have lot many bacteria, and we also know the roles of them in digestion. An interesting thing to know is that this is human in many of the species. You know, in horses and all, the things are a little different. Small intestines were found to have similar, or in fact little higher than the colon, the number of microorganisms in horses. So there might be species differences. Now what does they do? We know all the time that they are important for digestion. We all know by human experience that if there is indigestion, we take the probiotics to solve these problems. So these are very, very vital for indigestion purposes. But it's not just indigestion. Studies have shown, metagenomic studies, that these microorganisms from the gut and those in the gut epithelium can go to other organs, milk, liver, and other organs, and may even influence the brain function. They secrete short chain fatty acids. They secrete LPS, which can diffuse and reach all the way to the brain and affect the brain function. There's a bidirectional event, both for the liver and other organs, including brain. They may go and modify the brain function. Similarly, studies have shown that there's a stress or other condition which can cause the release of cortisol, say the hypothalamus pituitary axis. They may cause the release of cortisol. Cortisol will in turn lead to biosignaling pathway, lead to the increased transcription of cytokines, and these cytokines can again have function on the liver. Bacteria can direct, as we have talked earlier, the bacteria themselves can produce LPS, which can activate TLR pathway to activate the NF-kappa beta and NF-kappa B pathway, or inflammasomes to cause inflammation. They can also cause the release of the TLR4 pathway, particularly they can release the release of cytokines, and so the myoid differentiation pathway, MAP kinase pathway, Janus kinase pathway, and all can cause the insulin resistance, obesity. So studies have shown that the metagenomic profile of healthy animal, disease animals are different. Metabolic profile of animals with different feeding schedules are different. Metagenomic profiles of animals which are having higher feed intake is different. There's a specific population which are dominant, particularly related to the ratio of Bacteroides to Firmicutes. They are particularly, Bifidobacterium is another example. They can be useful. They can affect the feed intake. They can affect the growth rate of the individual. They can affect the food conversion ratio, very, very vital for the food animals such as pig or beef production in other countries. They're also very, very important for the health status of other organs. So we can use this, we can harness this potential of metagenomics to animal productivity. It can also be used for animal selection, say gut microbiota. You can analyze if you have two groups of pig or cattle which are known to have different different growth rate, for example. We know that research have shown, metagenomic studies have shown that they both have the profilings, and if you can profile that, okay, the gut microbiota or fecal microbiota of high performing or the fast fattening pigs are having this particular profile. So it can be used for genetic selection of animals, and in fact this is being now coming up as new area in metagenomic selection of animals for selecting the productive animal. A new area of the genetic selection is there. Studies have also shown that, metagenomic studies, that the genes associated with microbial resistance can dominate under certain conditions, and which can use to find out new antimicrobial strategies for treatment of animals. We know that fiber diets, suppose if you increase the fiber diet, a concentrate rich diet, it can change the microbiota, and then we can avoid to use that. If you are doing this kind of diet, probably we can have this kind of feeding schedule. See, you may require more of the fibrolytic bacteria if you feed is fiber diet. So that kind of strategies can be divided and can be used for better health and productivity of the animals can be done. And this is something which is coming up in animals, but in human, this is well known now. The functional food, the use of prebiotics, use of the probiotics, postbiotics, all are coming up into that. The lactobacillus probiotics, we all know it's very, very common nowadays. We do Yakult, that TV advertisement that comes, the yogurts, the curd that we eat, all those seems to have the pathways and how they can do. So if the microbial analysis of microbiome can help to identify newer probiotics that can help to increase the gut health, and by improving the gut health, they can have better impact in other organs, say brain function, tolerant to the stressful condition, mental health status, reducing the metabolic syndromes, particularly the insulin sensitivity, obesity, and so on. Liver function, liver cirrhosis, non-alcoholic liver cirrhosis cases are increasing a lot nowadays in our country. So it can have multiple utility in there. Now there we can keep on going these examples with gut microbiota. Rumen microbiota has also been studied, while earlier studies have shown not many populations, only 10 to 15% of microorganisms were recognized in the rumen, but now we know that many microorganisms are present. Wood profile is available for different species, cattle, goat, sheep, under different forage conditions, different dietary schedule, different parity level, and so on. So this keeps on going, and now we have many things to discuss microbiota, and that's all we can keep on going and talk. I think I'm ending up the time schedule with that. So the metagenomics has lots of studies, lots of advantages, both for the researchers to divulge new therapeutic strategy, preventive measure, diagnosis measure, improving the health status, animal selection, animal productivity, as well as to understand to gain the new insight into physiology and pathophysiology of several diseases, including the brain diseases, gut diseases, and different organs. But metagenomics also have challenges currently we are facing. One of the biggest challenges that we have is at every step, starting from sample preparation to DNA isolation to sequencing to post-sequence analysis, we have several things, and therefore the metagenomics has its own challenges. It's a developing field, and the data that we gain with this has to be carefully analyzed to avoid confounding interpretations and to get a robust results. Some of the things which is worth mentioning is, when you collect the sample, I have highlighted earlier also that when you do the sampling, you need the filtration of the unwanted animals. Say, for example, if you are looking for the bacteria, so you may try to leave out the viruses and all, so that you get only those results. But sometimes lysogenic organisms may be present in that the bacteria may get. So we have computational tool to do that. But computational tools may also present false negatives because of homolog sequences are not present in databases. When you do the DNA isolation, particularly if you're taking the DNA isolation from extreme environment, highly alkaline or highly acidic situation, then you will find that there will be DNA damages during the shearing damage to DNA, small site damage. The DNAs are difficult to then process. And if the DNA becomes smaller, you have to increase the sequencing coverage which again will require the computational power and all. Now, sequencing itself, we have talked earlier, is associated with the need for computational power, the need for speed, computational power, computational resources, particularly whole genome sequencing, because it generates gigabytes, even terabytes of data, depending on the sample types you are processing. There could be errors in assembly because there is interspecies similarity in certain sequences, and that's a very big challenge now. That's why a number of algorithms, number of softwares have been developed to handle this, but so far none has been able to solve this problem. Different algorithms will give different results. Now, if you are doing the OU2s and all proxy sequences, we know for certain species, but for many species, it's not known. See, 16S DNA you can use for bacteria, but many species not known, and we have talked about the limitations of this. And short read sequences such as Illumina sequences may identify the operons, may identify genes and all, but long operons or mobile insertion elements which are longer than 5 kb and all are sometimes difficult to identify with this one. And functional analysis, this is going on. This is a rapidly growing field. The functional analysis gives a lot of idea to us, but still it has its own lot of limitation. I think I'll stop my talk here. Thank you very much for your listening. If you have any questions. I'll be happy to take if you have any questions.
Thank you, Dr. Gupta. So if there are any questions, I request you, one or two people to, turn out, there's a raise hand option in there. You can click it and raise your hand, and we'll take a few questions, one or two questions, and then we'll move on to the next speaker because we are pressed for time. But if Dr. Gupta is available, then we'll take up all the questions together at the end. So if there are any quick questions, request for that. Anybody? So I hope all the attendees are able to,
There's one question: is databases resources available? So I think in the slide I have given, I think the question may have earlier. So different databases I have listed in one of the slides. We have silver, green genes, and so on. Rhizome, we have database, FACS database is there, PHACCS FACS database is there. I don't work in Rhizome, so I will not comment much on them. Yes, it is possible to do that. So this is something I do not work on Rhizome area, so I'll not be able to answer much. But there is a database, FACCOM is one of them.
If there are no more quick questions, we'll just move on to the next session, and then you can keep on typing your questions there. And Dr. Gupta, I request you to look at the questions and try to answer them there, or also later on if you are available, we'll after this session we'll have a talk because this is a shorter session. So next, before I'm starting the next session, I would like to let you all know that because of there's technical issues, Dr. Jyotsna was not able to present about India Alliance. So instead, we'll ask the next speaker, Dr. Madhavan Challa, to do the honors. I'll introduce you to Dr. Challa. So this session, the next session, and this introduction will be done by Dr. Madhavan Challa. He is a grants manager at India Alliance. So can we have the slide of Abdul? Dr. Gupta, if you can put your slide. Stop sharing your slide, Abdul.
Okay.
Thank you.
Can you confirm you can see my slide, Himanshu?
Yes. So the introduction slide, if you have, or that's fine if you. So I'll just go ahead with introducing Dr. Challa. So he's a grants manager at India Alliance. He has completed his master's degree in sciences from the Andhra University in Visakhapatnam and PhD from Anna University, Chennai. He then pursued his postdoctoral research at the Weill Cornell Medical College in New York and the University of British Columbia. His research work was focused on characterizing the proteins involved in transport across the cellular membranes, including connections and ATP independence and dependent phospholipidases. He has been associated with India Alliance since September 2016, and he would be a good person to introduce you to India Alliance because of his experience. So I now invite Dr. Chalat to turn on his video and okay. Just a moment, he's having some issues. In the minute, I'll request Abdul to share the slides.
Are the slides visible, Himashri?
Yeah, I can see the slides are up now. So now Dr. Chalat will introduce everyone to India Alliance. I hope I request him to turn on his video if possible. Thank you. Over to you.
I'm visible. Sorry about that delay. Thank you for having me here, and I hope everyone's doing well and having a great day. So it's a pleasure for me to talk about India Alliance with you all, to share our journey till now and what we have to offer to you in terms of funding opportunities, and also talk briefly about our contributions to the Indian biomedical research ecosystem. So briefly about me, like Himashri just introduced, I'm currently a grants manager at India Alliance. Next slide, please. So the basic remit of India Alliance is to support education, training and research in science and technology. India Alliance is founded jointly by Government of India through Department of Biotechnology and through a collaboration with a UK-based charity called Wellcome Trust. We also help build capacity in biomedical research to strengthen research ecosystems in India and to foster interdisciplinary international collaborations and make in an attempt to make science accessible to everyone. Next slide, please. So our journey started in 2009, though the talks started in 2000, talks and to form the alliance started in 2008. The first funding program from India Alliance started in 2009 with the launch of basic biomedical research programs for early career researchers, intermediate researchers and senior researchers. So we have 3 levels of funding for basic biomedical researchers: early career fellowships, intermediate fellowships and senior fellowships. In our through the way in 2013, with an eye to improve and encourage clinical and public health research in India, we also started the clinical and public health research fellowships program. Here also we have funding for early career, intermediate and senior fellowships. What these are and provisions of these different levels of fellowships will become more clear when I talk about the funding opportunities available from India Alliance. The second phase of India Alliance started in 2019 with the launch of 2 grants programs. So till then we were only providing research fellowship funds. Since 2019 we are also providing grants to researchers. There are 2 streams of grants that we provide. Clinical and public health research centers. These are aimed at building virtual research centers where researchers from different institutions or actually different institutions themselves can come together, collaborate, and try to address and solve an important health and clinically relevant research problem relevant to Indian context. And team science grants are interdisciplinary grants. They could be either basic biomedical or clinical in nature. These involve teams of researchers from different institutions or different departments. There should be at least 3 co-PIs along with a PI for this scheme. So since we are currently in the second phase of India Alliance's journey which started in 2019. Next slide, please. This is a distribution of where all our fellows and grantees are working currently. Till now, we have given 508 awards, which are being hosted at 145 organizations located in 47 cities in the entire width and breadth of India. As you can see, our influence covers the entire map of our great nation. Next slide, please. So I'll briefly talk about a few fellows who are currently involved in doing some very interesting research work in metagenomics area. So Dr. Ashwin Sainarayan Shesasai is our intermediate fellow hosted at National Center for Biological Sciences in Bangalore, and he investigates the studies global gene expression and evolution in bacteria using proteomics and metabolomic tools. Dr. Ambili Shivadas is a basic biomedical early career fellow hosted at St. John's University in Bangalore, and she studies integrative omics. She is developing an integrative omics approach to enable precision medicine for treating diabetes. Another early career fellow at JIS Institute of Advanced Studies in West Bengal. She is Dr. Kamakshi Surekha Paul, who is studying elucidation of CDAMP signaling pathways in mycobacteria. Next slide, please. Another basic biomedical intermediate fellow at IIT Bombay, Dr. Shobna Kapoor, studies lipid chemical biology and biophysics, and she's developing a platform to map the landscape of virulent lipid-induced effects. Dr. Arnabha Chakrabarti, another early career fellow who is hosted at Tata Medical Center in Calcutta. He's developing a hypothesis-based proteogenomic discovery approach for next generation applications in ALL. Dr. Akhilesh Pandey was our Margadarshi fellow. He worked on establishing a cancer center for molecular medicine. Next slide, please. Dr. Nishant Matange is an intermediate fellow at ICER Pune, and he is working to develop and identify drug targets for retarding antimicrobial resistance. And Dr. Shashank Tripathi, another intermediate fellow at IISc Bengaluru, is studying the role of host factors in influenza A virus pathogenicity on a genome-wide CRISPR-Cas9 screening platform. Next slide, please. So what does India Alliance offer to researchers in Indian biomedical research ecosystem? We offer fellowships and grants that help build research capacity in the different institutions in India and develop centers of excellence, and to catalyze internationally competitive research programs. Some of the provisions that help in developing these will be more clear when I talk about the provisions of our fellowships and grants programs. Next slide, please. In addition to our research funding programs, we are also actively involved in other areas to popularize science. Like I said initially, our mandate is to develop an excellent internationally competitive research ecosystem for India. In pursuit of that, we conduct science communication workshops where researchers from different institutions from all over the country are trained in different methods of science communication, grants writing, manuscript writing, how research ethics and modules like that. We also sponsor a workshop called Developing Indian Physician Scientists Workshop, where new clinicians who have not been involved actively in research are introduced to the different methodologies, and they get a little flavor of what goes on in research work. And so this is done with an objective of generating an interest among clinicians to become clinician researchers. Next slide, please. We also have lab leadership workshops. As you know, one of the important requirements for PI is to manage the lab. Along with their research, they should also have an ability to manage their lab effectively, manage their manpower effectively. So with this in mind, we collaborated with EMBO to organize leadership workshops for PIs working in different institutions of the country. Another step we have taken to popularize science in India is to award India Science Media Fellowships. These are given to journalists. So these are not researchers, these are journalists who write for many different mainstream media outlets on science related matters. And these were provided funding to go and do some field work on science related communications and science related news stories to highlight the research, main research developments happening in our country. Next slide, please. Along with this, we also organize research methodology workshops, especially for clinicians. This is not just doing research, but how to plan their research project, how to plan the ethics approvals, how to plan clinical trials and things like that. And this is done in collaboration with CDSI at THSTI. We have also launched something called India Research Management Initiative. This is aimed to strengthen the research management ecosystem in the different institutions in our country. We provide grants, fellowships, and travel awards to people who are in the area of research management. So this is to help free the time of researchers from the nitty-gritty of paperwork and bureaucracy of managing research. So a dedicated team of research managers can take care of that aspect of doing the research, of the research administration, while the researchers themselves can focus their valuable time on doing the real important stuff of research and don't have to waste their time and energy on paperwork and reports and things like that. Next slide, please. In addition to that, other public engagement activities include our partnerships with universities, public communication with school students, bringing awareness, and also partnering with organizations for webinar for public enlightenment. Next slide, please. So these are some of the in-house media and publishing efforts of India Alliance. We have a newsletter which is published every month, and you can sign up to this newsletter to get important updates and events about our fellows, their research, about the competitions that India Alliance conducts, about the funding opportunities from India Alliance, etc. We also have a podcast, monthly podcast called Science Together podcast series, and we also have science stories video series, which are both associated and are aimed at highlighting the research work of our fundees, grantees, and fellows. Next slide, please. So these are some of the handles where you can connect with India Alliance, either on Facebook, Twitter, Instagram, or on YouTube. If you want more information, you can write to us at info@indiaalliance.org. If you are interested in organizing workshops in association with India Alliance, please write to workshops email ID. And for any media related matters, please write to communications@indiaalliance.org. With that, we come to the end of this presentation and move on to the next part of my presentation on research funding opportunities at India Alliance. I hope everyone can see my screen, right? So we've covered these.
Yes,
So
Dr.
I'll skip to the next one. So India Alliance sphere of action, I've already described these to you, and distribution of our fellows. So what is the objective of India Alliance fellowship programs? So like I said, we have two programs, fellowship programs and grants programs. So I'll talk to you about them one after the other. So our fellowship programs are aimed at implementing globally recognized grants funding model, which is influenced by the different international funding agencies. We have collected the information, distilled them, and brought them to modified them to suit Indian context, and we are implementing. And in these 13 years of India Alliance's existence, our program has already been recognized as one of the best ways in which funds are managed in our country. We have also developed an effective mechanism to not just identify internationally talented, internationally competitive researchers based out of India, but we have also put in place mechanisms to nurture them into leaders in their respective fields. Our programs are also aimed at reversing the brain drain. Some of the mechanisms by which we do that, I'll talk about in a little bit. So we through these programs we enable people to come back to India who have moved out to do postdoc position or postdoc training, or for the PhD training. We help them come back to India and set up a base, find their footing in our country. We also provide a pan-India platform for biomedical researchers to interact and collaborate, and we have developed new mechanisms to promote public health and clinical research in the country. So we offer fellowships at for researchers at three different levels of their career trajectory. Early career fellowships are meant to launch the research career of a young researcher. This is aimed at people who have just completed their PhD and are planning to enter and start a postdoc training. Intermediate fellowships are for researchers to establish their lab and start a new research program. This is again offered to researchers with a little more experience than fresh PhD candidates. Senior fellowships are for leaders in their respective fields of research. These are more experienced researchers who will who are trying to expand their research programs and are trying to uh, uh, pursue innovative areas of their own respective fields. Like I said before, all these streams are offered for both basic biomedical researchers as well as clinical and public health researchers. The funding programs, one of the USPs of our funding programs is the emphasis on lead investigator. So the primary, as the name suggests, the fellowships, it is to identify the fellow who is talented and has the potential to become a leader in their respective fields. So there is a greater potential while scrutinizing the applications on identifying the right person for the particular program. Our fellowship programs also do not have any age-based restrictions. We do have eligibility criteria, but those are based on people's research experience. Like I said, our programs are aimed at people at 3 different levels of their research experience. So according to that, we have fixed eligibility criteria based on the research experience and not based on their age. Also, our funding programs are open to people from anywhere in the world as long as they're prepared to work in India and to help develop the research ecosystem in our country. So there is no nationality restrictions on applying for India Alliance funding programs. To help, especially to help, not just, I mean, this is not just for female researchers, but this is especially useful for female researchers who tend to take a break from their research career for family or personal reasons, with an objective to help such people. We also account for periods of non-research break in their career while calculating the eligibility criteria, so that we can help them come back into the research mainstream. Our fellowship programs come with a very generous personal support contribution from India Alliance. So anyone, even those people who do not have a faculty position in an Indian institution, can apply for our funding programs, and the institutions can confidently agree to host our fellows without having to worry about paying their salaries and things like that. In addition to that, our funding programs also have a 10% institutional overheads contribution, so that's an additional way to help the institutions host our fellows. All our applications and entire grants management system is done through an online secure online platform called ISS, India Alliance System. So there's no need for anybody to print things and mail it to us and things like that. Everything's done online. Especially I mean, though we had this system in place for the last 10-13 years, the utility of this system became very apparent during the last 2 years of pandemic, where people were not able to go out and post things and print things and things like that. Our fellows did not feel any pressure or any difficulty because the entire thing is online. Like I said before, also we have programs that are targeted at people who are aiming to pursue interdisciplinary research, and we particularly encourage people who are trying to pursue an interdisciplinary research program. So our funding committees, which help us select our fellows, their job doesn't end with just identifying the fellows. They are constantly involved in monitoring the progress and providing feedback to the fellows on their research programs. This again helps develop them into leaders in their respective fields. And it will become apparent in the next few slides that our funding program offers flexible and very generous, probably one of the highest in the country, amount of funding to pursue research. Again, like I said, to apply for India Alliance funding programs, information is available on our website, indiaalliance.org. Applications are accepted through an online system called India Alliance System, or ISS for short. If you are interested in applying for India Alliance funding, we highly encourage you to register and update and fill up your profile on ISS. Even if a funding program that you're interested in is not open at this point of time, it'll make your work easier when the particular funding round opens, so you can directly log in and submit an application. I'll now briefly talk about the provisions and eligibility requirements for our fellowship schemes. This slide shows the information about the basic biomedical schemes. The early career fellowship competition is open to researchers who are in the last year of their PhD program, and those who with up to 4 years of post PhD research experience. So the eligibility is from -1 years to +4 years of post PhD research experience. The total fund available, budget available for early career fellowship is 1.7 crores, which includes up to 10.36 lakhs per year contribution as personal support for the fellows. So like I said, if people who do not have a faculty position or a permanent salary support from an institution in India can also apply for India Alliance funding programs. We will provide full salary up to this specified cap specified in this slide. For example, somebody has a salaried position at a host institution, and if the salary is lower than this salary cap that we have fixed for early career or intermediate or senior fellows, we will make up the difference in the form of a top-up. And if somebody has a salaried position for which the salary is higher than the cap that we have fixed, that entire amount can be used as research costs. For early career fellows in basic biomedical research, they can even request a support staff in the form of one technician from the second year of their fellowship. Like I said, early career fellowships are supposed to be postdoctoral research, so the expectation is that most of the research is done by the fellow themselves, and so this is not like a supervisory role. So the fellows are expected to do the research themselves and find a lab where they have major equipment and facilities to conduct the proposed research. So with this in mind, there won't be any support for purchasing major equipment, but there will be support for purchasing consumables, chemicals to access facilities or for other miscellaneous expenses. One of the interesting features of our fellowships, especially for early career and intermediate fellowships, is a provision called work outside host institution. What this does is this allows the fellows to visit the lab of a collaborator or a mentor who is outside their host institution. This mentor or collaborator could be either within India or abroad. And so the fellows, early career fellows, can spend up to 2 years of their 5-year fellowship in work outside host institution. And to make it more attractive, while if somebody is going abroad to pursue work outside host institution, in addition to their normal personal support, we will provide additional $3,000 per month of subsistence allowance. Okay, so this is not instead of the personal support. This is in addition to the personal support. So along with all the other costs, we also provide contingency fund up to 2.5 lakhs for 5 years. We call this flexible funding allowance. This can be used for any unexpected expenses that might arise, that for which the fellow may not have considered in their application. We also provide 7.5 lakhs of travel allowance, or called travel to meeting allowance. This is to help the fellows register for conferences, travel to conferences, accommodation while they travel to conferences, or to visit their collaborators' lab for a few days and things like that. So this is 7.5 lakhs for early career fellows. For intermediate and senior basic biomedical research fellowships, we have a floating eligibility criteria of 4 to 15 years of post-PhD research experience. So it is expected that people at this stage would know where they fall in terms of competitiveness and their career trajectory, and they can choose which level of fellowship they want to apply to. The budget cap for intermediate fellowship is 3.6 crores and 4.5 crores for senior fellowship, and salary is 13.82 lakhs for intermediate fellows and 16.12 lakhs for senior fellows. The work outside host institution provision is not available for senior fellows. However, if they need to travel abroad for collaborative visits, they have the option of requesting funds under a different head called miscellaneous head. This could include travel costs as well as subsistence costs for their stay and food and things like that. Unlike the early career fellows, intermediate and senior fellowship applicants can request support staff, and the support staff can include PhD students and postdocs and technicians, lab assistants, administrative staff, everything. They can request up to 2 support staff positions on an intermediate fellowship application and up to 4 support staff positions on senior fellowship application. The travel allowance for intermediate fellowship is 7.5 lakhs and 10 lakhs for senior fellowship, and contingency is 7.5 lakhs for intermediate fellowship and 10 lakhs for senior fellowship. These are the provisions for clinical and public health fellowship schemes. Mostly the provisions are similar with minor changes, and you'll see that the budget and personal support contribution are slightly higher for clinician researchers. So this is because we provide non-practicing allowance. So for people with a clinical degree, MBBS, MD, MS, they are given a non-practicing allowance, which is not available to people with a basic PhD degree. So for clinical researchers, the budget cap as well as salary support is slightly higher. Another difference between clinical schemes, clinical and public health scheme, and the basic biomedical schemes is that we have a single floating eligibility criteria for clinicians and public health researchers. This is to encourage clinicians to participate and compete for the fellowship programs. Again, people with up to 15 years of research experience can apply, post-qualifying degree research experience can apply for early career or intermediate or senior fellowship schemes. So briefly about our competition cycle timeline. Screening for our fellowship programs happens at 3 stages. It starts with a preliminary application, when we make the competition and application form available on our ISS platform. The preliminary application consists of the applicant's CV and a brief abstract of their proposed research work. These applications are screened by the office, and all the competitive eligible and applications are invited to submit what is called a full application. Full application consists of a more detailed research proposal, more detailed budget. Preliminary application doesn't include the budget, which is present only in the full application, and other details about collaborators, mentors, work outside host institution, and things like that. The full applications, each full application is reviewed by at least 3 international experts in their respective fields. The second level of scrutiny of the fellowship applications happens after this stage, when the selection committee goes through the applications and shortlists the most competitive and most eligible applications to the interview stage, which is the third level of scrutiny. After the interview, which is usually face-to-face, a few of the applicants are awarded the India Alliance fellowships. So this entire process takes about 10 months from the preliminary application stage to the award declaration stage. There are a couple of policies that need to be highlighted in this context. So the first is our resubmission policy, according to which if you are invited, if an applicant is invited to submit a full application and the application is reviewed by our the peer reviewers, external peer reviewers and our internal selection committee, that will be the only opportunity for the applicant to apply to that particular scheme or to a scheme that is junior to that. Like I said, we have 3 levels, right? Early career, intermediate and senior level fellowships. So if somebody submits a full application to say intermediate fellowship scheme and their full application gets declined either at interview stage or at shortlisting stage after being reviewed by the selection committee, the applicant will not be able to apply again either for the intermediate level fellowship or to an early career fellowship. So that will be the only opportunity. However, they are free to apply to a senior fellowship if they wish to. Okay, so that is our resubmission policy, and we are very strict about this. So please, if you are interested in applying for our fellowship program, please think carefully about the timing when you are going to apply, and whether you are going to be competitive by applying to that time, or maybe you will be more competitive applying at a later time with better research track record and publication track record. Another policy that I would like to highlight is the dual fellowship policy. So India Alliance, like I said, provides very generous salary support. So if you receive India Alliance fellowship, you will not be eligible to hold any other fellowship at the same time. So if you have another fellowship like a Ramalinga Swami fellowship or a Ramanujan fellowship or a NPDF or something like that, you're welcome to apply. We will consider the application, but if you get awarded before you can activate our India Alliance fellowship, you will have to decide whether you want to continue with the India Alliance fellowship or continue with the other fellowship. You can't have both at the same time. So now, what makes an application? What does the office, what does the selection committee look for in an application? Obviously, from your application, from the research proposal, it should be clear that the applicant has ownership of the research proposal. That is one of the important, it can't be derivative, it can't be continuation of somebody else's work. It can't be continuation of your postdoc work or your PhD work. It has to be novel. It has to be an important biomedical program. It has to be a novel biomedical problem, and the applicant has to have complete ownership of the research proposal. The committee also looks for the feasibility of the research plan. Often we see that people have very good ideas, but it should also be practical. It's not enough that the idea is good, but it should be doable, right? So you may have a very great idea, but if you do not have the expertise or the tools or the collaborators or the manpower to execute that particular research proposal, then it's not going to be competitive in the field. We also look for scientific productivity of the applicants. This could be demonstrated in the form of publications, presentation record, oral presentation, poster presentations, mentoring, especially for people applying to senior fellowship, experience mentoring PhD students and postdocs, filing patents, and other parameters used to measure scientific productivity. Of course, we also look for motivation of the researcher. Like I said, our program is to build an ecosystem, a research ecosystem, and to identify leaders of Indian science. So we look for people who are going to stick to the research and who are motivated to pursue a career in research. Uh, Again, like I said, all our applications are reviewed by international peer reviewers. So it is important that the applications have a good grantsmanship, especially bad grantsmanship would be unclear timelines, unclear experimental plans, spelling mistakes, bad grammar. So if very often in our country, English is not our first language. So there is no harm in asking for inputs from somebody who has good grasp of English, or your mentors or your seniors. We also look for supportive letters from your supervisors, who might provide recommendation letters. So these letters shouldn't just be that I support this application. It should be clear why this application is good, why this person is good, what are their research accomplishments, what is their motivation, what is their track record, and things like that. And if you're going to have mentors, mentors should explain what their contribution to the fellowship program is going to be, how are they going to help in your career growth and things like that. And then if you're going to have collaborators, collaborators should clearly say what is their contribution, what are they bringing to the table in terms of research contribution. So these are some of the high level points on what makes an application strong. Like I said, biomedical, the early career fellowship competition happens once every year. The next call, if you are interested, will be in July, August, and with interviews in November, in May, June 2022. Next round of basic biomedical senior and intermediate fellowship applications will be accepted again in June, July, August 2022. Clinical and public, we have just a couple of days back closed the application round for this year's clinical and public health research fellowships. The next call will be early next year with interviews in November 2023. So for more updated information, for more current information on competition launch dates and competition timelines, it would always be a good idea to visit our website or to sign up to our newsletter, so you'll get updated information. Another stream of funding that we launched in 2019 is the grants screens. Like I briefly mentioned earlier, these come under two main heads: team science grants and clinical and public health research centers grants. There is a third embedded sort of a program called clinical research training program. This is not a separate program, but this is embedded within the clinical and public health research centers program. Team science grants are intended to help researchers, or they are actively aimed at researchers who want to pursue interdisciplinary and collaborative research that addresses an important health issue in our country. So this should involve at least 2 PIs from different institutions, and the research should be, as a rule, interdisciplinary in nature, and should aim at a large very impactful research problem. Clinical and public health research centers are aimed at establishing virtual research centers that aim to improve clinical and public health research ecosystem in India. So this should include training, other administrative structures, training of students, training of research, staff, things like that. In addition to research, okay, and one of the components of CRC grants program is the CRTP program, where the applicants can request support to recruit physician scientists to train them as part of the CRC research program. So like I said, people from at least 2 different research institutions and at least 3 minimum PIs, including 1 lead PI, have to be present as part of the team science grant team. And people should have at least, the main PI, the lead PI should have at least 5 years of experience independently leading a research team. Like all our programs, there is no age or nationality bar, but the research has to be performed. The lead PI should be in an Indian institution. The collaborating PIs, the co-PIs, can be anywhere in the world, but the lead PI should be in India. In fact, in this program, we actively encourage researchers to find collaborators from institutions outside India. Again, as part of one of our emphasis on developing international collaborations. The total amount of funding available is up to 10 crores, and this can include staff, equipment, and all other requirements of a research program. Similarly, for clinical and public health research centers also, applicants can request up to 10 crores for 5-year duration. But these, unlike the team science grants, these are to be requested by institutions. These are institutional grants. So at least 2 institutions can apply for clinical research centers grants, and the lead institution has to be based in India. So again, in going with our emphasis on developing biomedical research ecosystem in India, the lead institution has to be in India. The collaborating institutions can be outside India also. The CRTP program is for physicians who want to be trained to become clinical researchers, and this is embedded in the CRC program, like I mentioned earlier. And funds can be requested in each CRC grant application to hire up to, or to train up to 12 CRTP fellows. And there will be a fund, the support available in addition to the 10 crores that will be that can be requested for CRC program. Up to 50 lakhs per CRTP fellow. Additional funds can be requested in the CRC application. In addition to these are the main research funding opportunities available from India Alliance. We've already discussed these other opportunities that are available. And again, once again, the information on how to get in touch with us. Feel free to write to us anytime on our info@India Alliance email ID. We are happy to provide any information that you would need to apply for our funding programs. Thank you for your time.